Dear learners, I am Ramya, working as Assistant Professor in the Department of Biotechnology, St.Josephs College of Arts and Science for Women, Hosur. We are going to learn about bacterial growth curve and its significance. How we can grow the bacterial culture. When a broth culture is inoculated with a small bacterial inoculum, the population size of bacteria increases, showing some pattern that can be obtained as a graph. That is what its distant curve is called as bacterial growth curve. Method of obtaining bacterial growth curve. A population growth curve for any particular species of bacterium may be determined by growing a pure culture of an organism in a liquid medium at a certain temperature. Samples of the cultures are collected at fixed intervals, that is for every 30 minutes, and the number of the viability of an organism is determined. It can be plotted as a graph paper. This diagram shows the growth curve. The initial stage is lag phase and the growing stage is called as exponential phase or log phase, and then at one stage the bacterial growth will be stopped, that is called as stationary phase, and then the multiplied bacterium slowly destroys, that is called as death phase. So this four stages we are going to see in detail. The bacterial growth curve shows the following four distinct phases. First one is lag phase. After a liquid culture broth is inoculated, the bacteria require some time for its multiplication. It prepares itself for multiplication. The time between the inoculation and the beginning of multiplication is known as lag phase. During this phase, there is an increase in size of bacteria, but no appreciable increase in number of bacterial cells. The bacteria act or cells are active and metabolically. The duration of lag phase varies with the bacterial species, nature of culture medium, incubation, temperature, etc. The second stage is called as lag phase or exponential phase. This phase is characterized by rapid exponential cell growth, that is 1 to 2 and 2 to 4, and it will be divided into 4 to 8 and so on. The bacterial population doubles during every generation. They multiply at their maximum rate. The bacterial cells are small and uniformly stained. Growth rate will be greatest in this log phase. When plotted on logarithmic graph paper, the log phase appears as steep sloped straight line. Next phase is called as stationary phase. After log phase, the bacterial growth almost stops completely due to lack of essential nutrients, lack of water, oxygen, change in pH of the medium, etc., and accumulation of their own toxic material metabolic wastes. It is during this phase that the culture is at its greatest population density. The death rate of bacteria exceeds the rate of replication of bacteria. Endospores starts forming during this stage. Bacteria becomes gram variable and shows irregular staining. And also it produces exotoxins. The last phase is decline phase. During this phase, the bacterial population declines due to death of cells. The decline phase starts due to accumulation of more of toxic products and autolytic enzymes, and the nutrients will not be there. It will be utilized by the microorganisms during its multiplication. Involution forms are common in this stage. Some cells assumes various shapes, becoming long filamentous rods or branching or globular forms that are difficult to identify. Some develops without a cell wall and are referred to as protoplast, spheroplast or L-phase variants. When these involuted forms are inoculated into a fresh nutrient medium, they usually revert to the original shape of healthy bacteria. Why we are studying the bacterial growth curve? Why it is important to understand the bacterial multiplication? The study of bacterial growth curve is important when aiming to utilize or inoculate known number of bacterial isolate, for example to enhance plant growth, increase biodegradation of toxic organics, or produce antibiotics or other natural products at an industrial scale. Growth kinetics is also useful for accessing whether particular strains of bacteria are adapted to metabolize certain substrates such as industrial waste or oil pollution. Bacteria that are genetically engineered to clean up oil spills, for example, can be grown in the presence of complex hydrocarbons to ensure that their growth would not be repressed by the toxic effect of oil. Today's question is, what is the other name for log phase? And one more question, why we have to study bacterial growth curve? Thank you, dear learners.